Hello, everybody. Welcome back to the RYD show with your host, John Kiriru. Today, my guest has requested to remain anonymous due to fear of being doxxed by the medical technology industry. You may remember his former employer, the once proclaimed innovative health technology company Theranos. Some may remember them for their $9 billion valuation and promising mission statement. In comparison, however, others recognize them as a company whose founder sits in prison for defrauding her investors of over $400 million. Without giving away his identity, I can tell you today's guest has a PhD in pathology and laboratory medicine. He has worked as a head researcher at Theranos since its founding, and today he is a professor, researcher, and author with the focus of his work being on preventative healthcare. Getting blood drawn is a common practice in today's healthcare system as it helps us find out how well specific organs are functioning or provide diagnoses of diseases. However, not everyone is as fond of the needle that is stabbed 2 inches deep into your arm to extract the blood as others. Expect to learn why, though, in theory. Theranos had a great idea, but a terrible product, including, how the tests were done, how the tests stack up against the gold standard, and the nitty-gritty of what happens to your precious blood after it's being drawn, and much, much more. So, Doc, thank you for coming into the studio today.
My pleasure, man. Been a long time listener.
Back in the early 2010s, just bring, blast to the past. I remember seeing billboards and advertisements for Elizabeth Holmes being coined, as the reincarnation of Steve Jobs. Such a high praise, you think, for anyone in Silicon Valley, let alone such a young college dropout. So, what was Holmes' goal, motive for the company that habilitated so much praise, all this worship from economists?
Our mission was to find a new way of blood testing that didn't require a butterfly needle.
Mhm.
Once our second round of investing had ended, I saw the company's values slowly shift towards providing an inexpensive and effective method to test blood.
Right.
Elizabeth used to picture our Edison testing machine in every household. In theory, the Edison was meant to provide quick and precise results of one's blood with just a singular drop. Essentially, Theranos aimed to make blood testing less painful and more accessible. Looking back, investors weren't putting money necessarily into the company or even in the product, but they were more so betting on Elizabeth.
Right. Right.
She had this aura that entranced anyone willing to listen.
Yeah, I can tell. I mean, she did promise, a machine capable of running all these wide range of tests with just a few droplets of blood. So, just to help me understand this, why wouldn't more companies have invented this earlier if such a thing was possible? Think of like, a way to make a universal testing machine that was smaller, cheaper to produce, and could be, mass distributed. Don't you think Pfizer, Johnson, heck, even Siemens, would have been all over this by now? Did Theranos make any significant breakthrough in that realm of technology?
We promised a lot to the public for sure, but we failed to meet their expectations because our techniques, machines, and tests were not as advanced enough for the time to do multiple tests with just a few drops of blood.
Hmm.
The design of the machine consisted of microneedles and a computer chip that would analyze blood results. Unfortunately, this wasn't possible with the small amount of blood that we were limited to because Holmes had promised that this would be possible. A small amount of blood was not adequate because some of the tests required a more significant amount of blood
Right.
because there may not be enough concentration of a particular protein. For example, a testing device would need to accurately detect a deficiency of a particular protein,
Hmm.
and this could tell if a patient had a deficiency of some
Right.
sort or any health concerns in a specific amount of blood.
Hmm.
Then it could calculate and extrapolate outwards based on the composition of the patient and total volume of blood in their body.
Okay, so especially in a medical testing machine, before putting that product into the market, you definitely need a seal of approval from the FDA. So in your case, each test you claimed Edison had passed, right? So what issues did you guys run into trying to get this device approved? Any green lights potentially?
As more investors boarded, our product deadlines were shortening. Shorter deadlines meant less testing and more errors to an already error-prone testing device.
Hmm.
To meet the investor stated deadlines, we used a hacked version of the Siemens Advia Centaur XPT, which is amino assay system. Uh, and this was used to run our tests that we reported as being taken from the Edison.
Hmm.
The Edison was so flawed in design that the blood had to be diluted in a 1 to 20 ratio of blood to water, resulting in unreliable results.
Right.
I knew this was malpractice from the beginning, but I couldn't turn away at this point in my career. As for FDA oversight, we were in a very peculiar case since we didn't send tests to a third party
Hmm.
lab to run analyses. Instead, we had our own CMS certified lab and didn't sell our analyses to other labs, making us exempt from any FDA oversight.
Hmm.
However, bringing the product to the mass market was another story.
So which tests were approved?
One test that the FDA approved was a test for herpes.
Okay.
This test consisted of using Western blot and STS page, which is a type of Western blot.
Hmm.
This aids in measuring whether a specific protein is expressed in a sample, figuring out the protein's abundance and the protein's size. In this case, the Western blot was used to confirm the purity of the desired GG2 antigen. Western blot tests are best used for herpes because they have a high sensitivity, and can help distinguish between HSV1 and HSV2 antibodies.
Mm-hmm.
The high sensitivity may be one of the reasons why the herpes test was able to conduct on the Edison.
Right. That makes so much sense. So, tell me about these methods that, you guys use. How are each of these tests conducted? And, why didn't you guys opt out for a different type of test?
Let me put it to you this way. Of around 200 tests, only one was FDA approved. Now, let's say a company was to find a way around FDA approval or find a loophole in the semantics of regulation. This would open up the possibility of making claims that far exceed supporting evidence.
Okay.
For example, we ran trials in Arizona through a cancer clinic, and many patients reported receiving positive tests confirming their cancer had returned while in remission.
Mm-hmm.
In reality, these were false positives. Many of what Theranos couldn't offer were considered laboratory developed tests, which bypassed pre authorization from the FDA.
Expand on that.
Unlike authorized approval, these lab developed tests are self validated, with the lab paperwork only being reviewed during routine inspections.
Okay.
We used STS-Page and a laboratory test for detecting antibodies called enzyme linked immunosorbent assay, commonly known as ELISA, to analyze antigens and antibodies separately.
So let me get this straight. This test you called ELISA, right? And you mentioned this test was done on antibodies. So let's delve deeper into this. Can you identify like what specific antibodies? Like, can this also be run on other cell components as well?
Mhm. So Theranos' machine, the Edison, was designed to perform immunoassays, which look for an antibody or antigen in blood or fluid.
Mm-hmm.
The machine would look for different antibodies depending on the test being run. The protein type in antibodies is a globulin protein called immunoglobulin.
Right.
These immunoglobulins play a crucial role in the immune response by recognizing and binding to specific antigens, such as bacteria and viruses.
Mm-hmm.
Therefore, when diagnosing patients for diseases, illnesses, and deficiencies, immunoassays such as ELISA and STS pages are used to identify the antibodies in the patient's fluids, such as blood, to identify the root cause of the problem.
Of course. So how is, traditional tests, different from the so-called ELISA test that you guys did?
To detect the presence of bacteria and viruses, traditionally the ELISA test was one of the most crucial ways the biomedicine and research industry involved the detection of specific antibodies in the blood.
Mm-hmm.
So, for better understanding, the antibodies are formed by specialized white blood cells.
Right.
The STS page is traditionally used to separate mixtures of proteins. An STS page assesses a protein sample's purity, protein expression evaluation, and immunochemical identification and quantification of proteins.
Right.
However, some limitations of the STS page include needing help to assess enzymatic activity, protein binding interactions, protein cofactor detection, and RNA and DNA detection. The ELISA assay uses catalytic properties of enzymes to detect and quantify immunologic reactions. These assays are used in clinical analyses.
Mm-hmm.
ELISA is also used to detect antibodies to viruses
Right.
and other antigens in serum or blood. Some diagnostic tests that ELISA can conduct are the presence of antibodies, level of tumor markers, estimating hormone levels, tracking disease outbreaks, detecting past exposures, and detecting drug abuse. However, one limitation of ELISA is that if preparation is not done effectively, it can lead to false results.
Can you give me an example on that?
Mhm. So, the Theranos team conducted a study on expressing and characterizing a soluble form of recombinant mature HSV2 glycoprotein G for use in anti
Yeah.
HSV2 IgG serodiagnostic immunoassay.
Mouthful, man.
It really is. So, in case you didn't know, HSV2 is herpes simplex virus type 2, which is transmitted sexually. And the HSV2 specific glycoprotein is used as the antigen for serodiagnosis
Mm-hmm.
of HSV2.
Mm-hmm.
This study used 234 clinical samples,
234.
which 134 were high risk samples, including 45 samples from pregnant subjects, and a panel of 100 mixed diagnoses samples.
Yeah.
STS page analyzed the purified antigen, and then ELISA was performed on the purified GG2 antigen by serial dilution in a buffer.
Okay.
The ELISA assay indicated that including the complete extracellular domain of the mature GG2 antigen does not lead to cross-reactivity in HSV1 patients in an indirect ELISA serological assay.
So what's the difference?
The indirect ELISA has a higher sensitivity when compared to the direct ELISA.
Okay.
The findings showed a few negatives in the HSV2 positive samples and even more false negatives in the HSV1 and 2 positive samples. The report stated that the GG antigen maintained high performance in patient sera despite the false negatives. This study is among many of Theranos' fraudulent studies or reports.
Okay, yes. So I've, done one of the reports, right? And so once Theranos got exposed, how did that kind of impact the general public, do you think?
If I recall, at this point in time, there were a couple companies, including Pfizer and Walgreens, who had pooled in large sums of money. And so, since these two companies were publicly traded, their investors were the one who were eating the loss.
Mhm.
And so, this was due to the CEO's poor judgment. But ultimately, Theranos was forced to pay back all the money they cheated their investors out of.
Okay.
Walgreens knew about these concerns from the beginning, but they feared that their competitors might come in and swoop them up. And so instead of stopping, they opened up 46 testing centers around America. And since these 46 testing centers were open to the general public, many people reported receiving false positives. And so these false positives
Wow.
of something like cancer led to the patient having a heart attack or a stroke due to stress.
Yep.
And this stress was not only felt by the patients themselves, but their friends and family as well. So this is no joke.
Of course, never. Yeah. So, yeah, thank you, Doc, for coming to the studio today. It was just really lovely speaking with you today.
My pleasure.